Assalamualaikum dan salam sejahtera. This is class for General Biology. I'm Dr. Shaida and I will be your instructor for this course. We are in Chapter 2, Cell Division and Reproduction under the topic of mitosis. Have you ever wondered how our nails grow? Moreover, the characteristic of our newly grown nail resembles that of our previously trimmed nail. What is the mechanism that leads to the growing nail? To answer this, we need to understand about the human genetic profile. A human consists of genetic materials that are stored in the cell nucleus and mitochondrial genome. The number of nuclear genome in human cell is 46 linear DNA molecule, which is known as chromosomes. It is built from 22 homologous chromosomes and a pair of sex chromosomes which are denoted as X and Y chromosomes. Humans have two basic cell types, which are the somatic cell and the germ cells. Somatic cells are formed during asexual reproduction by mitosis, whereas germ cells are formed during sexual reproduction by meiosis. Germ cells of humans are known as sperm and ovum, involved in the synthesis of haploid gametes that are used in sexual reproduction, whereas somatic cells that include the stem cell, fat cells, neuron cell, red blood cells, and all other cells are engaged in the construction of multicellular human bodies. The somatic cell is known as adult stem cells. It includes all types of cells in the human body except the sperm and the egg cells. It is able to divide and duplicate its DNA during the mitosis, resulting in two identical cells. In an adult, humans have more than 3 trillion somatic cells. Somatic cells are diploid, produced by mitosis in asexual reproduction. Two sets of homologous chromosomes are seen in the nucleus. Somatic cells are differentiated broadly into 4 main types of tissues, which are connective tissues, epithelial tissues, nerve tissue, and muscle tissue to organize into a complete human organism. A gamete is a reproductive cells of an animal or plant. In animals, female gametes are called as ovum or egg cells, and male gametes are called as sperm. Ovum and sperm are haploid cells, with each cell carrying only one copy of each chromosomes. During fertilization, a sperm and ovum unite to form a new diploid organism. The germ cells undergo meiosis to produce haploid gametes, which include the sperm and the eggs. The development of a new progeny organism is then initiated by the fusion of these gametes at fertilization process. Back to the question of how nail can grow. The nail is a plate-like, keratinous, translucent structure that consists of highly specialized epithelial cells. These epithelial cells is somatic cells that conduct the mitosis to generate new cells for development. The nail needs to continually growing to replace cells that are dead and repair the damaged cells. The cells must replicate or make identical copies of themselves. In order to do this and maintain the proper number of chromosomes, the cells of eukaryote must undergo mitosis to divide up their DNA. The dividing of DNA will ensure that both the old cell parent and the new cell, or known as daughter cell, will have the same genetic makeup. Thus, it will make your new grown nail to be similar as your previous trimmed nail. The idea or term of mitosis was first introduced by a scientist named as Walter Fleming in 1882. The term originates from Greek word mitos, meaning warp thread. Mitosis is the process by which a cell replicates its chromosomes and then segregates them, producing two identical nuclei in preparation for cell division. Thus, mitosis provides equal division of the cell's content into two daughter cells that have identical genomes. Let's go closer to the process that happens in the mitosis. Before the cell is ready to divide, it must complete several important tasks. It must grow, copy its genetic materials, and physically split into two daughter cells. Cells perform these tasks in an organized, predictable series of steps that make up the cell cycle. The cell cycle is a cycle rather than a linear pathway because at the end of each go-round, the daughter cell can start the exact same process over again from the beginning. The stages of the cell cycle are divided into two major phases, which are the interphase and the mitotic or M phase. During the interphase, the cell grows and makes a copy of its DNA. During the mitotic phase, the cell separates its DNA into two sets and divides its cytoplasm, forming two new cells. Interphase is the first stage of the cell cycle that happens before the cell division. During this phase, the cell matures and prepares to divide and copies its chromosomes and centrioles. The interphase is a preparatory phase for the cells to divide. Cells will create complete copies of DNA for themselves. As closer look to interphase, it is actually consists of three phases which are G1, S, and G2 phases. These three stages are preparation steps prior to mitotic division to occur. The G1 phase is called as the first gap phase. During the G1 phase, the cell is quite active at the biochemical level. The cell grows and accumulates the building blocks of chromosomal DNA and the associated proteins, as well as sufficient energy reserves to complete the task of replicating each chromosome in the nucleus. The cells grows physically larger, copies organelles, and make the molecular building block as it will need them in the later steps. The S phase or synthesis phase of interphase takes the longest because the complexity of the genetic material being duplicated. Throughout interphase, nuclear DNA remains in a semi-condensed chromatin configuration. DNA replication results in the formation of identical pairs of DNA molecule, sister chromatids that are firmly attached to the centromeric region. At the center of each human and animal cells, the centrosomes are associated with a pair of rod-like objects, which are known as centrioles, which are at the right angles to each other. The S phase involves duplication of microtubule organizing structure called the centrosome. The centrosome helps separating the DNA during M phase. The centrosome will give rise to the mitotic spindle, the apparatus that orchestrates the movement of chromosomes during mitosis. The centriole will help organizing the cell division. These centrioles, however, are not present in the centrosomes of other eukaryotic species such as plant and most fungi. During the second gap phase or G2 phase, the cell grows more, making more protein and organelles and begin to reorganize its content in preparation for mitosis. In the G2 phase, the cell replenishes its energy stores and synthesizes proteins necessary for chromosomes manipulation. Some cells' organelles are duplicated, and the cytoskeleton is dismantled to provide resources for the mitotic phase. There may be additional cell growth during the G2 phase. The final preparation for the mitotic phase must be completed before the cell is able to enter the first stage of mitosis, the G2 phase, and when mitosis begins. The process of mitosis or cell division is also known as the M phase. This is where the cell divides its previously copied DNA and the cytoplasm to make two new identical daughter cells. By the end of M phase, the sister chromatids separate from the original chromosomes and form a new cells. The M phase is broken down into four subphases, which are the prophase, metaphase, anaphase, and telophase. Prophase is the first stage of mitosis. During prophase, the nuclear envelope of the cell begins to break down. If it is an animal cell, the centriole separates and each moves to the opposite end of the cells. As the two cells' centrosome move toward the opposite poles, network of protein fibers that are made of microtubules gradually assemble between them, forming the network that will later pull the replicated chromosome apart. These protein fibers are known as spindle fibers. As the centriole takes place at opposite sides of the cell, they send out more spindle fibers. These spindle fibers seek out the sister chromatids that are present in the cells. Spindle fibers from one side of the cell attach to one of the sister chromatids. The spindle fibers from the other side of the cell attach to the other sister chromatids in the chromosomes. They attach at a point called the kinetochore, which is a disc of protein that is on each side of the centromere. The spindle fibers will move the chromosome until they are aligned at the spindle equator. In normal human somatic cell, it consists of 46 chromosomes. During metaphase, each of the 46 chromosomes will line up along the cell of the metaphase plate. Spindle fibers align the chromosome along the middle of the cell nucleus. This line is referred to as the metaphase plate. This organization helps to ensure that in the next phase, when the chromosomes are separated, each new nucleus will receive one copy of each chromosome. During anaphase, the centromere splits, allowing the sister chromatids to separate. Spindles attached to the kinetochore begin to shorten. These exert a force on the sister chromatids that pulls them apart. Spindle fibers continue to shorten, pulling the chromatids to the opposite poles. This will ensure that each daughter cell will get identical sets of chromosomes. The kinetochore spindle fibers shorten, allowing for the 46 of the newly freed chromatids to be dragged to one end of the cells and the remaining 46 chromatids to be dragged to the opposite end of the cells. These separated sister chromatids are known from this point forward as daughter chromosomes. At the conclusion of anaphase, each of the cells has an identical and a complete set of 46 chromosomes or 23 pairs of homologous chromosomes that are still diploid. The telophase begins once the chromosomes have completed separating and reached the opposite poles of the cells. During telophase, the network of spindle fibers is dismantled. The chromosomes decondense, and around each complete set of chromosomes, a nuclear envelope will begin to form. At this point, the actual process of mitosis is actually completed. Cytokinesis is the final stage of the cell cycle in which it can occur anywhere from anaphase into telophase once the chromosomes have separated. The mechanism involves splitting of the cytoplasm, which causes the cell to divide roughly in half. These two halves are identical copies of one to another. The newly splitted cells is a fully functional cells in which each is known as daughter cells. After the completion of the cytokinesis, the cell cycle for the somatic cell is now complete and each daughter cell begins the process anew. In summary, mitosis is a type of cell division in which one cell or the mother cell divides to produce two new cells or known as the daughter cells that are genetically identical to itself. Mitosis consists of four basic phases, which are the prophase, metaphase, anaphase, and telophase. The process of mitosis generates new cells that are genetically identical to each other. And mitosis helps the organisms to grow in size and repair any damaged tissues. That's all for today's class. Thank you for listening. See you in the next lesson. Bye bye. Assalamualaikum warahmatullahi wabarakatuh.